We are live. I know it didn't connect again.
Hey followers, safety first. I hope you like that candle. I won it at Soils Trivia Night. Awesome. I have a script. Hello and welcome to Live from the Nursery event. Funnily enough, we are actually not at the nursery. But before we get going, let me introduce myself. My name is Josh Nuzzo, and I'm the nursery manager for Point Blue Conservation Sciences STRAW program. If you're not familiar with STRAW is an acronym, and it stands for Students and Teachers Restoring a Watershed. It's the education and outreach program of Point Blue, where we get students out onto the landscape to do real habitat restoration. And as the nursery manager, I grow the vast majority of the plants that students are planting on the landscape. Behind the camera, we have the Jastrola, our nursery technician. Hey.
No.
Yeah.
Hey.
Nice. And yes, so you may have noticed we're not at the nursery. It was scheduled to rain, but it's not. So here we find ourselves at our Petaluma headquarters in Sonoma County. This is where we're all based out of, although Point Blue is international. This is where we're set in Marin and Sonoma County. So our aim for these live programs is to engage nature lovers in the SF Bay Area and beyond. Lost my spot. In habitat restoration efforts led by Point Blue and our many partners to increase your awareness and excitement about the ecological importance of ecosystem restoration. So today we're coming at you live with our program, part of our Live from the Nursery series, and this one's titled No Flowers for Valentine's Day, which is just a really it's me and Alishka's brainchild, but what we're going to be talking about today is doing propagation when you don't have seeds and you don't have any seeds or flowers or fruits, because things are always going on at the nursery and we're always growing plants. And I just want to show you all a couple of things we do in the winter time and for when we don't have the normal genetic material, the seeds, the germoplasms, am I right? So I guess before we get going, I just want to do like a few little introductory things. So there's two different kinds of reproduction. There's sexual reproduction, which is where two gametes meet and create a genetically unique individual. So that's like, for plants, that's what yields a seed. And then there's also asexual reproduction, which is cloning, where you're taking material from a parent plant and turning it into multiple plants. So what we're going to be displaying today is asexual reproduction. This is something we do quite a bit at the nursery. It's not our ideal, because ideally we have seeds to yield genetically unique individuals. having genetically unique individuals reduces our risk of bringing pests and pathogens into the nursery. It also creates a more resilient restoration project for disease, and also provides more flexibility in the face of climate change. But we don't always get the seeds. So sometimes we don't get seeds because maybe the seeds were delicious and wildlife got it first. Maybe it was just a scheduling logistic thing. We just didn't get out there. You know, there's a series of microclimates in the Bay Area. Even within Petaluma, we're finding there's differences on the west side and the east side. And then also sometimes plants don't invest a ton of energy in seed production. Their life habit might be like growing by rhizome, or maybe their seeds just aren't super viable. I don't know. So it's good to have all these different little tips and tricks in our pockets. So with that being said, we're going to be talking about asexual reproduction, and there's some other little things I'd like to point out to you as well. If y'all remember back to bio class, then you'll hopefully remember this exact drawing. This is just a generic plant, and I like to, when we teach this to the students, I like to just draw this anyway, just as a fun reminder of the different parts of the plant. So like starting from the bottom, we have the root structure here, roots and the root cap and the taproot. We're not going to be dealing with the roots today, but it is interesting to note my nails, and also the root cap, because the root cap, just like at the node and some of this above ground material, contains the apical meristem, which is like the stem cell of plants, and that's what we're going to be manipulating today in one of our ways of doing propagation. So just moving up the plant, we have our branches. Sorry, I had to label it because these are opposite branches. You'll notice on our other drawings sometimes they're alternate, which every other. This one I drew opposite, and that's important for what we're going to be doing today. We also have leaf, flower, and then terminal bud is something that we're going to be wanting to pay attention to as well. Yeah, so just another zoomed in example of what we're going to be doing. This is a really accurate depiction of the California rose, or Rosa californica, one of the plants that we can do vegetative propagation with, and one of the plants we have here today. You'll notice some of the things are the same. This is a compound leaf made of a series of leaflets. Super fun. You'll also notice here, just like our generic plant, we have a terminal bud, and then what we really want to be paying attention to is these nodes. And node is just like almost like a generic word, just noting any point of growth on the plant. So anywhere that there is a leaf, a branch, a flower, or just a bud, that is a node. Node, 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 terminal bud and node. And the space in between, I have okay handwriting. The space in between the node is called the internode. Awesome. And the reason why I'm giving you all this information is because what we're going to be doing with the plants here is we're going to be manipulating the node. If you remember just seconds ago, I was mentioning the apical meristem material. That can be found in every single node and on the tips of roots. And the apical meristem can be manipulated naturally, but then also by us to yield more plants. So just like us, plants are controlled by their hormones, right? Hormones tell our bodies what to do when. They affect like our mood and like our growth and where we are in our life cycle. And plants are no different than us. They do have a different suite of hormones, and I just like rattled off a few here. Yeah, you might be familiar. If I were to guess that you were familiar with any of them, I would imagine it would be ethylene, which is what they use, that plants emit to ripen their fruit. You know, if you ever did that like fun experiment where you put a banana in a bag versus a banana on the counter, banana in the bag ripens more, that's an effect of ethylene. So that's one that might be familiar to you all. The hormone we're going to be using today is the cytokinin, which allows plants to grow organs. And it's located in the meristem. And when we say organs, we generally mean roots and shoots. Awesome. Take a moment. Sweet. Okay, so now that you're all experts at plant biology and physiology, we're going to get to the good stuff. So one of our ways we like to reproduce plants when we don't have any seeds or the plant isn't producing seeds or like we forgot to get out there, is what we like to do is we like to do things from cuttings. You all might be familiar with this because it's the way a lot of people reproduce their houseplants. You know, you trim your little vine and put it in the water. This is very similar to that. But something that's a little bit different than our houseplants is we really care about where the plants are coming from because these are going for habitat restoration. So when we're looking to clone plants, we're trying to keep plants from the area. You know, oftentimes we're restoring areas that maybe are super degraded, but we like to keep things generally within the watershed. So when you're selecting plants, you want to make sure there's a few little guidelines that I think we talked about last time, similar to seed collection. But for cuttings, it's slightly different. One thing is you want to collect early in the day. You know, that'll help plants retain water. It'll help them just like their longevity in general. Right now we're looking at this beautiful sticky monkey flower. And just like with seed collection, we never want to be collecting more than 5% of the total plant. You know, we don't want to have, we're obviously having some sort of impact on the plant itself, but we don't want to have like such a big impact that it'll change its trajectory. And then we don't want to collect more than 5% from the individual, and then never more than 25% of the total population of plants. We also are looking for things on the individual parent plant. We want to make sure that it doesn't look diseased or sick. Things that might tell me that it's looking diseased or sick if the plant's leaves are discolored, if there's any sort of pests accumulating around the plant, and then if there's any like crazy die-off, like if like half the plant is dead, maybe that's a no-go for us. And then we want to, because we're going to be cloning plants and they're all going to be genetically identical, we want to make sure that we're collecting from as many individuals as possible to kind of get some sort of genetic diversity into our restoration site. So from here, we have 4 different genetically unique individual monkey flowers, so we'll collect from all 4 of them. And then it's also worth noting when you're collecting that there's going to be death in every step of the way. You'll collect, some of your cuttings will die. You'll process them, some of your cuttings will die. You'll pot them up, some will die. And then, I don't know, then they'll just die just for funsies afterward. But that's worth noting because, like, say you want 25 plants, and just knowing what we know about this process, you should probably collect more than that. There's an equation here that I'm not going to rattle off to you, but there's a method to this. You always want to, although you want to be conservative and reduce, like try to minimize your impact on the parent plant, you still want to be collecting more material than the plants you want at the end of this process. Excellent. So now that I've said my piece about our ethics and some collection guidelines, let's get to it, right? So I'm putting on some gloves because today we're going to be working with first the California Wild Rose, or Rosa californica. It is a fabulous plant for habitat restoration. You can see it creates these really charismatic, beautiful pink flowers, which are fantastic for all sorts of pollinators. It also creates, from the flowers, it creates these beautiful red rose hips, who are fantastic sources of, food source for animals. And then another fantastic thing about it, it's just so fantastic, is that it spreads rhizomatously. And that's a word I'll be using a bit today. A rhizome is basically a modified stem that grows laterally underground, and then another plant will pop up. If you all are familiar with strawberries, you know, you'll have one strawberry plant, then it'll shoot out like a little tentacle, and then another one will grow, and then it'll create a patch. A rhizome basically is the same as that, but underground. And so that's really fantastic when we're working on our degraded landscapes because it stops erosion, it provides really good habitat, it also provides food for a variety of species, and it adds complexity to the restoration site, you know, by creating this kind of like hedge of shrubs. So now that I've sold you on the rows, I'm going to select one of our cuttings. They're so sharp! These ones were collected from our nursery, from the grounds itself. So what you'll want to do, we collected a really long sprig because I really wanted to illustrate what we were talking about earlier with the plant anatomy and physiology. So if you'll notice here, it is covered in spines. I'm wearing these gloves. Oh boy, they go through the gloves. I'm not bleeding, but I am in pain. And so something I also want to point out to you are these little bits here and here. So unlike our generic plant that I drew on the whiteboard, this one has alternate leaves. And what I'm pointing to is the buds. So, I mean, yeah, the buds, but also the nodes. So we have node, internode, node, internode, node, internode, node. And there, you'll notice that there's quite a variety of like length in the internode. And that's worth noting, depending on what you're doing. You know, you don't want a cutting that's like super long or maybe one that's super short. I don't know, each plant is different. And although maybe these are the same species, each branch will be different due to like abiotic factors like weather or moisture or sunlight, things like that.
Before we begin, do you mind if we do one with vegetation? The nodes are maybe hard to see on the camera.
Oh my
and maybe
God.
one with the
leaves.
the one with the leaflets might be easier to see the nodes.
Oh my God, yeah, sorry about that. The nodes can be cryptic.
Nodes can be cryptic, ain't that the truth?
Ooh, this one's got somebody's random hair. Beautiful.
Yeah.
Awesome.
Yeah, perfect example.
Yeah, so sorry about that, followers. But yeah, here, I guess it's a little bit more illustrated. Like I said before, the node is any point of growth along the stem, and that can be another branch, it could be a flower, or it could be a leaf. So here, the nodes are very clear because they're all these compound leaves. And here you can really see alternate. It's like zigzagging up the plant. And you'll also notice here, like I was saying earlier, that the space, the internode, is different for each one. Yeah. Awesome. Thank you. Yeah, if there's anything else like that, just let me know. So what we're gonna need, we collected our sprig from the wild, and there's gonna be a few materials you're gonna need. If you have garden shears or Felcos, fantastic. If you don't, fantastic. Scissors will work too. The arts and crafts kinds, or kitchen scissors. I have a lot of choices today. And then because we're worried about pests and pathogens, it's also nice to have some 70% isopropyl alcohol and just spray off our little Felcos. Other things that you'll want are we have this beautiful melange of perlite and vermiculite. Perlite is like a naturally occurring mineral. It's also processed and sold to us. It adds air and space to any sort of like potting mixture. I'm sure if you're familiar with soil, you'll see the little white things in that. That's the perlite. And then also in here we have a mix of vermiculite, which is another mineral that actually is like little sponges that hold on to water. And today we're going to be doing this in perlite and vermiculite only because that's what we like to do at our native plant nursery. But you could do this like in peat moss mixture. You can add peat. You can add some normal soil. You can add like coconut coir. Whatever you've got is good. Just note that we're going to be chopping this plant up, so it's going to have a lot of different like wounds or areas for entry for pests and pathogens. So what we like to do at the nursery is we like to use the perlite and the vermiculite because it's naturally sterile. So there's less likely a chance of there being, ASMR.
Oh, wow.
We know they like ASMR, the people online. So using this kind of media allows us to kind of reduce the chances that something will infect our plant and kill it. But everything dies. Okay. So what we're gonna do, we got our stuff. Oh, another thing you'll want is the rooting hormone. You can get this at any garden store. I like this one because it comes out as like a powder, which is a lot easier to use, but there's also liquid hormone. You could also make your own hormone, doing like extract of willow bark. I've tried it, didn't work for me. Doesn't mean it won't work for you. This is the stuff I like, it's just really easy to use. And this is the human-made manufactured set of kinin. And this is the hormone that we're gonna use to manipulate the plant. Another thing you're gonna want is some vessel to put your plant in. Today we're using a just like a regular pot, but I've grown them in Tupperware. I've actually grown them in this very container before, and I've grown them in pots and containers. And then I have a finished product I'll show you at the end. So what you're gonna do, I was like, I lost my stuff. Is what I like to do. I like to prepare my pot first. I just switched these boxes up. They look exactly the same. It's just the perlite and vermiculite, but I added water to this one. It makes it a little bit easier for me to use. Scoop it up in there. And then I press it down really tight. Because although we had all this, we added the perlite to have some air. We don't like to have a ton of gaps in there. We want to have the plant have contact with the media. So you just want to press it down really, really tight. Just like so. And now we're ready to grab our shears or Felcos or pruners or scissors. I don't care. And then we're going to be looking at the nodes again. I'm freaking obsessed with nodes these days. And what we like to do is, like I said earlier, each plant's going to be a little bit different. But one rule that we like to keep the same is we want to have a minimum of 4 nodes. Also, we want to not be so close to the end here because this is like a fragile new growth point, and it'll generally die back anyway. So I'm going to do a little bit of this. You'll notice that I cut right at the node there. If I left any space, like if I cut in the middle of the internode, that tissue there is going to die and then it'll rot. And then there's potential for the whole cutting to rot. So I cut right at the node, and then what I'm going to do is I'm going to count down a few nodes. So 1, count with me, y'all. 1, 2. No, I was joking. 3, 4. Okay, and so I'm noticing that the inner nodes here are really, really small. Everybody has their personal preference. I like my cuttings to be a little bit bigger, so I'm going to go 5, 6, 7, 8. It's 4 a.m. and I'm wide awake. Okay, and then I cut, you'll notice I cut right below the node there. And I'm going to cut, hopefully you can see this, I'm going to cut at an angle. These are dull. You don't have to do that, but I like to do that because it increases the amount of surface area, and that's where we're going to be applying the hormone. Also, we want to make sure part of this is going to go into our perlite vermiculite melange. And I like to so some of these are going to go underground. So I'm going to just clean it up a little bit. I'm going to chop off their leaves. It feels cruel, but honestly, it's not. Don't worry about it. But you'll see what I'm saying. I was talking about earlier about how there's going to be lots of open wounds. Oh, and my Felcos are too chonky. So I'm going to use some scissors to get right in there. We want to expose some of the nodes. Get right into that apical meristem material, right? Whatever. So this is what we would call a process cutting. If you're worried about where you're going to be growing it, if it's really hot or something and you think the leaves will just die off, maybe I'll cut a few more. It's less material for the plant to support in this weakened state. Awesome. So now we have, this is what we like. Let's turn this into a whole other rose bush, huh? Rosa californica bush.
Beautiful. And can you remind us why we cut off the top of the cutting?
Yeah. So we cut off the top because as you can see on this one, this is where there's lots of new growth. The tissue is really, really tender. And what I found just by trial and error is that if you leave this, oh, these gloves are a little dirty, don't look. If you leave this material on here, it's really, really flexible compared to the center material. And it's just gonna droop down, and then it's gonna die. And then it's gonna rot, and then the mold and stuff could spread to the rest of your cutting. So we'd like to do a preemptive measure and just chop the top, just like this, making sure that there's a node on top so new growth will spread, just like that. Yeah, thank you. Yeah, if there's anything else that comes up, I'm kind of like blasting a ton of info at you. If there's anything else that comes up, please interrupt. So right here we have our rooting hormone. And you'll remember that we cut at the bottom at an angle to increase the surface area. And then what we're gonna do is, get ready, everybody. You put it in the rooting hormone. I like to smear it around. The rooting hormone is a little expensive. I think it's like $8.99 at Friedman's, which is basically like a nicer Home Depot. But I'm using that sweet nonprofit money so I could get as much rooting hormone as I want. So I like to be really liberal with the application. I know some people are like more conservative. I like to glob it on. And you'll notice I'm trying to cover the whole node. If I had more time and like was a more patient person, maybe I would cover the second node with rooting hormone too. But I think for the sake of this, we'll just cover the bottom node. And you just want to make sure that that whole node and the open wound is completely covered. And then something I forgot to show you all is one of my favorite tools to use in the nursery. It's a metal chopstick. So helpful, so easy to clean. And what we're gonna be doing here is you wanna make sure that you create a hole. Each node's gonna be different, so I can't tell you a specific way, a depth about how you wanna be installing your plant into the media. But we do know that we've chopped 1, 2, 3 nodes leaves off, so we're probably gonna be going like about that deep, which is a couple of inches, a few inches. So I make the hole first, and then I place the cutting in there, and then I like to tuck it in with my little fingers, just like so. And there it is, folks. That is it. And so what we do at the nursery is we'll, once we potted it up like this, we'll put this in the greenhouse where it gets regular misting, and just to ensure that the media stays wet. But if you're at home and you're doing this, you could do this on your like counter or like your windowsill or wherever. It doesn't need to be in a greenhouse necessarily for you to have success. You just want to make sure that it doesn't really dry out too much. I've never tried this, so I don't know if it's a bad idea, maybe putting a tray. I don't know. I've seen what people do is sometimes you put it in the whole thing in a gallon Ziploc bag, and that creates your own kind of greenhouse where it's not necessarily sitting in water, but you'll see like dewdrop. Yeah, but yeah, cool. So I think before we move on to another one.
Yeah, and it's 12.
Oh shit.
But Lishka has a great question about why is it not centered in the pot? And I think to answer Lishka's question, you are super welcome to center it in the pot. This is just one cutting, but when you look at the branch, you could easily make 4 or 5 more if you wanted to. So to maybe save on medium space, or save on medium, instead of putting each one in an individual pot, you could put them all in the same pot.
Exactly, that's so true. And yeah, sorry, there's a lot going on with asexual reproduction. No flowers on Valentine's Day. But yeah, V's so right. We can make many, many cuttings out of this. And then just before you go, if you have time, we were going to do cuttings of the monkey flower too. You can notice that just like the rose, it's got this nodes, internodes, the leaf arrangement or the branch arrangement is opposite. I was going to do like a whole like potpourri of different plants. And we can chop this up too. And just like the roses, cut the branches off and then expose the node to the rooting hormone. And we do it the exact same way. So I guess this is how we do it in the nursery. Yeah. And we can do like hundreds per flat like this. And we actually have quite a few of these. This is our beautiful snowberry, the Symphoricarpus albus, which is a plant that like creates berries, but not a lot of viable seeds. So we largely produce it through vegetative propagation or cloning.
Yeah, and this is, to answer Lishka, this is a great example of why we wouldn't maybe place one in the center, because we can, it's kind of hard to tell on here, but you can like really just like keep maxing it out.
Yeah. And I'd say you can even fit more in here.
Yeah, and I like to imagine that all the roots are trying to talk to each other.
You're like, why did this happen to me?
I feel like they have better success when they're you have more in a pot for some reason.
Yeah.
I don't know why.
I feel like I read a study about that. Yeah, I guess we're out of time, y'all.
Yeah.
So thank you so much for tuning in. I really appreciate your attention.
Yeah, we have one more question if you want to answer before we leave.
Oh my god.
There's a question about when you're applying the rooting hormone. Is there a preferred way of doing it, with a Q-tip, or how?
Yeah.
What's your preferred way of applying the rooting hormone?
If I were at home and no one was watching, what I would do is I would stick it straight in the bottle. And that's really nice for me. We don't do that here at the nursery because we want to make sure that there's no cross-contamination. But honestly, sometimes I usually wet the bottom of the cutting, and then the powder will just glob right on. I mean, you could do a Q-tip. Mm-hmm. I don't see why not. It might be a little bit tedious. So I like to just like stick it right in.
Awesome.
Yeah. Awesome. But yeah, thanks for tuning in. We really appreciate it. We're going to do more of these, and we're going to be better about our timing because there's so much to say about plants. But yeah, feel free to comment, like, and subscribe. And yeah, keep an eye out for more things happening on social media and like in the wild world of Point Blue. Oh yeah. And then this is a snowberry that's been produced by cutting. It used to be a little stick, and now look at it. It's like a real plant.
Awesome. Awesome sauce.
Happy Valentine's Day, or not. Live your life. Bye.